Started. So today we are going to move into Chapter 2, the Biology and Behavior chapter. I've got the advanced organizer for you listed below here. So here's how we're going to approach this biology chapter. We're going to begin today by looking at the tools of the neuroscientist. The neuroscientist is just a scientist who studies the brain and maybe the connections between the brain and different aspects of behavior. So a biological psychologist sometimes might be referred to as a neuroscientist, or they might say that they study neuroscience. Then we'll look at the major parts of the brain today. We're going to be looking at 7 major structures and then 4 major parts of the overlying cerebral cortex. That's going to be the major focus for us today. Then as we get to the end of our face-to-face class session on Tuesday, after running through all the major parts of the brain, we will finally then zoom in to the cellular level, taking a look at the cellular components of the brain, that is the neuron. We'll look at the structure of the neuron. Usually that comes up at the very end of the face-to-face class session on Tuesday. And then we run with the rest of that and a focus on neurocommunication in next Thursday's online session. So online today on Thursday, doing major parts of the brain, continuing that, zooming into the cellular level on Tuesday face-to-face. Next Thursday online, talking about neurocommunication. If you had AP Bio in high school, you probably heard of something called the action potential. We'll be talking about the action potential next Thursday in our online session. So we are 12 days away from test number 1. I'm giving you up in the upper left corner here the breakdown for the 40 questions on test 1. I do this before every test to give you some sense of how to budget your time. Okay, so the introduction section that included some of the major theories, the history of psychology, some of the subfields within psychology that we did on the first 2 days, the last segment of our first face-to-face, our first online session, and then the balance of our first face-to-face session. 7 questions there. The research method slides, those were the maroon ones last Thursday, and then on our face-to-face get-together on Tuesday. 14 questions will come from there. And as I mentioned on Tuesday, the vast majority of the questions in terms of the breakdown here, we're most heavily weighted towards the biology chapter. So almost half of the questions here coming from Chapter 2 on biology. You will have your first set of learning curve quizzes due on Chapter 1 this coming Sunday night at 11 p.m. Do not wait until the last 20 or 30 minutes to try to do those quizzes. There will be 3 sections you'll have to do, 1A, 1B, and 1C. They will be worth 4, 3, and 3 points respectively. So remember, always 10 points per chapter. How the points break down depends on how many quizzes there are in the chapter. Most chapters have 3. There are some that have a little bit more, a little bit less than that. Questions about any of that regarding the preparations for test number 1? As I mentioned on day 1 online, one of the very best things that you can do to help yourself learn is to distribute your studying over time. We're 12 days out. We do the first test on Tuesday, the 16th of February, so it's a great idea to get rolling with your studying now, or maybe over the weekend. Okay, if you're planning to get at all this on Monday night, the 15th of February, right before the test on the 16th, that's not going to work real well, I promise you that. All right, so the only other thing I want to mention as I paste the learning outcomes into the chat box here is that we take a different approach to the biology chapter from the textbook. The textbook likes to zoom in to the cellular level first and then zoom out. I think for a number of reasons it makes a lot more sense to do what we're doing, which is to begin zoomed out looking at the major parts of the brain and then say, okay, now that we've learned about the hippocampus, let's zoom in and look at what goes on in there at the level of individual cells. So the consequence for us in terms of this learning outcome list in the chat box is that we're really focused on initially the bottom few. So we're really going to be working on numbers 9 and beyond, 9 through 13 this morning. And then we'll circle back up to the cellular level as we go forward. Okay, so they're in the sequence they're in because it makes it consistent with how they're laid out in the textbook. But just make a note that we start big and zoom in whereas the textbook starts small and zooms out. All right, we're going to begin this review of biology and behavior by talking about the tools of the neuroscientist. Basically, we're going to be talking about these 5 tools that scientists use to study the brain. And the approaches include looking at post-mortem brain specimens, looking at EEGs or electroencephalograms, looking at PET scans. PET stands for positron emission tomography. Looking at MRIs, magnetic resonance imaging, or maybe even doing an fMRI, a functional magnetic resonance imaging technique. And in fact, it is a strange looking thing, but the acronym for fMRI is little f, capital MRI. It's a little bit strange, but that's the way it works. Questions about the lay of the land here. Does this make sense? We're going to run through these 5. This is one of my overview slides that lets you know kind of the map that we're going to be following as we take a look at these different techniques. All right, so let's begin with a look at post-mortem brain specimens. Hopefully you're not eating because we've got a nice juicy brain to look at here. The post-mortem brain specimen approach lets us look at the brain and do what is called a gross examination. I'm not saying that to be cute and make it a pun, but it is an overall rough examination of the brain. And just to orient you, I'm going to tell you that the front of the brain is over here on the left. The back of the brain is over here on the right. That would then mean that this is the top of the brain. I could just type top, T-O. P is what I'm trying to do there. And then this would be the bottom of the brain down below. Okay? So what is this? Again, a rough examination of the brain. And because it's post-mortem, what do we know? Well, we know that the person is deceased, right? The person has died. So in terms of giving us useful information to help an individual and maybe improve their daily functioning, this is not the way to go, right? This is something that was probably a more important method of study maybe 100 or 150 years ago. Might still be used today when we're looking at someone in terms of some kind of autopsy and examining maybe what was going on in their brain that might have led them to behave and do the things that they were doing. It does give us a really nice look at the brain's overall structure, okay? And really the main thing I want to point out here that's obvious as you look at it is when we're looking at the brain, what we have in the outer shell here is this series of kind of nooks and crannies and these bumps and ridges and valleys and so forth. I want to label those for you and tell you that those are known as the cerebral cortex. And what's cool about the cerebral cortex is that it's where most of the really interesting stuff that we want to study as scientific psychologists. It's where things like reasoning and planning and abstract thinking are going on. Personality, things like intelligence are mediated by the functioning of this overlying cerebral cortex. So the way I think about the cortex is it's effectively like the shell or the bark, if you will, that's wrapped around underlying structures. We're going to talk about midbrain structures and hindbrain structures. Well, those underlying parts are wrapped in this bark or this casing or this shell that's known as the cerebral cortex. And before we finish up today, in our last 15 to 20 minutes, we'll be taking a look at 4 major lobes or sections of this overlying cerebral cortex. Okay? So this first approach to studying the brain that neuroscientists might use is admittedly pretty limited in the good things it can offer to us. Okay? Gives us a nice overall look at the overall structure of the brain, but not a lot of information, obviously, about things underlying the overlying cerebral cortex. If you're curious about this brain and taking a look at it for a second, you might say, well, this looks kind of weird. This section in here, like, there are some pretty significant parts that appear to be missing. Well, yeah, there are. And so this would be the left hemisphere, right? If the brain is oriented, as I've said, that this is the front over here, and this is the back over here, we're looking at the left hemisphere of this brain. And if you ask me to guess maybe what this person's functioning might have been like, well, I would say, especially given the damage that we see right in here, they would have had problems with language comprehension. Now you might say, how in the world can you know that there are problems here with language comprehension, given just this quick overall look at the brain? Well, it's because this part of the brain in here, in the top rear part of what's known as the temporal lobe, is called Wernicke's area. And we know that Wernicke's area has to do with our ability to comprehend what other people are saying to us. So if we had a way to look at your brains right now, as you're listening to me giving this incredibly interesting lecture about parts of the brain, we would be able to see activation in this Wernicke's area as you're listening to language. It turns out that there's a corresponding area kind of down in here that is involved not with language comprehension, but it's language production. So if we had a way to look at my brain right now, because I'm yammering on at you for 75 minutes straight today, we would see activation in the part of the brain called Broca's area, named for Paul Broca, who first identified this part of the brain. Okay, it's at the bottom rear part of the frontal lobe, and we'll say more about it before we wrap up today. But I just wanted to be able to point this out to you and say, these are the kinds of things you could see with this gross examination of a postmortem brain specimen. Okay, good enough. Tool number 2 in our toolbox is the electroencephalogram, the EEG. Let's begin by noting that it is a recording device. So these electrodes that are connected are recording devices on this woman's scalp here. We are not delivering shocks. This is not electroconvulsive therapy. We'll talk about that in the final segment of our course. This recording device is simply measuring something. And what it's measuring is electrical activity. I sort of think about it as the electrical hum, if you will, that is going on at the level of the overlying cerebral cortex. So this is great. We can connect this right back to what we just saw right here on the postmortem brain specimen. All of this bumpy stuff is the overlying cerebral cortex. And what we're saying here about the EEG is that this is the level at which it gives us information. The EEG, in terms of its limitations, doesn't give us information about underlying brain structures. Not going to tell us anything about what's going on in the midbrain, right in the middle of our head, or any hindbrain structures that sit below that. Okay, so for each of these tools in the toolbox, what I want you guys to understand is what they can do for us, and then where are the limitations, right? Where does this technique or this tool bump up against limitations, things that it doesn't show us? Well, there are really 2 limitations to the EEG. Number 1 is that it doesn't give us a brain image, right? Look at the monitor over here. Is that like a picture of a brain? No, that's just the recording of what's going on. So the first limitation of an EEG is that it's not an imaging technique. And then the second limitation would be the fact that it can only give us information about what's going on at the level of the cerebral cortex, right? So its maximum depth of penetration, if you will, for telling us something about what's going on in the brain is just at the overlying shell, right? Only at this level. If there's something underlying that overlying cortex that you want to get information about, the EEG is not the tool for you. What does it tell us? What are these wave patterns? There's all these squiggly lines. What the heck is that? Well, the EEG wave patterns, as it says here on the slide, correspond to different states of consciousness. So let me give you a couple of examples here. Give you a brain wave tracing for being awake and alert, and I'll give you a corresponding one or one to contrast about what goes on when you are in deep sleep. Okay? So let's take a look here. When you are awake and alert, you get these brainwave tracings that look something like this. They are small amplitude, relatively high frequency brain waves. I'll tell you what that means in just a second here, okay, after I get done tracing this masterpiece. So they are small amplitude, which in English means the height of the wave from top to bottom is pretty small. And they're high frequency. That means there's like a lot of them passing by a given point in a given amount of time. We can contrast that awake and alert brainwave tracing from the EEG with one that we might get from your brain at, let's say, 2 or 3 o'clock in the morning. What we're going to get then is large amplitude, relatively low frequency brain waves, right? We're going to get these giant brain wave tracings that look like this. So the amplitude here again is from the top of the wave here to the bottom of the wave down below. Clearly that's a much bigger amplitude when you are in deep sleep at 2 or 3 o'clock in the morning than right now at 9:45 when you hopefully are awake and alert. Okay? So who might use the EEG? Somebody who is studying sleep and dreams. There are sleep researchers who use EEGs a lot. EEGs can actually be used in looking at different kinds of seizure disorders, things like epilepsy. For example, the EEG might be part of the toolkit that you use in studying that specific problems. Okay? So we could check off the first couple of our tools here in the toolbox. We can look at the brains of people who have died. We can do EEGs if our question is about something going on at the level of the overlying cortex, and we don't need a specific brain image. If we want to get images and pictures, now we can turn to the third tool in our toolbox, which is the PET scan. Again, PET stands for positron emission tomography. And this technique is really interesting because it involves taking the patient and injecting them. What do we inject them with? We're injecting them with a tiny harmless amount of radioactively tagged glucose. Now you might be saying, why in the world, what are we doing here? Well, what we're doing when we're doing a PET scan is we're taking a look at where the fuel goes. Here's the logic. Let's tag the fuel, kind of like put a tracer on it, right? A GPS chip, if you will. Not literally, I'm just using that as an analogy, right? We put this tracer on the fuel, and the logic is let's follow the fuel, and where the fuel gets metabolized or burned up, that is going to be a part of the brain that is more active. And on our image here, with this brain imaging technique, we're going to see reds and oranges for areas that are more active. So I like to think about these as the hot colors. Whereas down below, we're going to have inactive areas appearing on the image that we get from the PET scan. They're going to be lighting up as blue. Okay, so these are going to be the cool colors. What we really like about the PET scan is that it gives us great information about brain function. Okay, what it doesn't give us is it doesn't give us great info about what? About brain structure. Okay, we don't get a beautiful look at the major parts of the brain in terms of their overall structure from the PET scan. So let's take a look at the image over here on the right. This is from the UCLA School of Medicine. It is a PET scan output when someone is being given different sorts of auditory stimulation. So they might be listening to something being read, they might be listening to music, they could have maybe words on the screen as they're looking at or listening to the story or listening to the song. What we've got here is a look down into the brain. So to orient you here, so things make sense, let me tell you that this is the front of the brain. If that's the front, that means that this is the back. And if we're going to keep being consistent, then this would be the left hemisphere, the left side of the brain, and this would be the right hemisphere. And that's the same orientation for all 4 quadrants here. The resting state, language, music, and then language and music together. What we're getting here effectively is a Goodyear blimp view down into the brain, right? So during the Super Bowl on Sunday, we'll have the Goodyear blimp up there showing you the football field from above. Now we're looking, in this case, down into your brain. So a slice through here called a horizontal slice on the image. And effectively what you have to do is think about the upper left quadrant here as the baseline. Okay, this is you in a resting state in the PET scan machine. And after we get that baseline in place, we can look at the changes that occur when you do different activities. So on the lower left side here, we're processing language. Tell them in the chat box, what areas look like they're becoming more active because they're red and orange as you're processing language or listening maybe to words being said into your ears here in the PET scan machine. Where do you see activation going on? Some in the front, some in the back. Tell me about the left versus right. Is it more on the left or more on the right? Yeah, more left. So one of the things that we could say here is in terms of lateralization or hemispheric specialization, we're seeing more language on the left. So I'm going to type in here, left equals language. Okay, and that's a nice little mnemonic device here because both left and language begin with the letter L. Oops, I'm trying to circle it. And I'm circling it in black on a black background. That doesn't work real well. So left and language, right? You guys are right. There is some activation in the frontal lobe compared to that baseline up above. That's probably because the person is focusing attention on some stimulus. So the frontal lobe, as we'll learn, has to do with attention and sort of locking on and paying attention to something. There's also activation back here, which makes me suspect that maybe what was happening was the person was looking at words on the screen that were maybe also being read aloud to them. The reason I say that is it turns out, as we're going to learn, that visual processing, vision, is processed in the back of our head. Crazy as that sounds, right? You got eyes in the front of your head, but at the level of the cortex, where the visual processing goes on is actually in the back of the head. Look at the music, lower right quadrant. What do you see? More left hemisphere activation or more right hemisphere activation? Where are the reds and oranges here? Yeah, we're going more to the right side there. So it looks like maybe as you're listening to music, the right hemisphere is more active in that kind of an activity. And then the upper right, I really think, is sort of a combination of what we've got down below on the lower left and the lower right. So we've got frontal lobe activation, we've got some activity in the back, probably because of vision, and we have both left hemisphere and right hemisphere activation happening there. Okay? Questions on that? Do you see what you get and what you don't get with a PET scan? So you get function, you don't get structure. You want structure? I've got something for you. Look at the beautiful picture of the brain here from the MRI. The right hemisphere was more active as you're listening to music. So the MRI uses magnetic fields and radio waves to give us this beautiful image of brain structure. So this is kind of the reverse of the PET scan, right? The PET scan gives us function, but not so much structure. The MRI gives us beautiful pictures of structure, but not so much function. What's this overlying stuff right here that we saw on the postmortem brain specimen? What do we call that? The cerebral cortex. Awesome. Underlying the cerebral cortex, we've got a structure here that we'll be discussing at the beginning of class on Tuesday. It's called the corpus callosum. It ends up that it's the bridge between the left hemisphere and the right hemisphere. Okay. Then we're going to talk about some other underlying structures here. We'll talk about the thalamus today. Here's going to be where we get down into the pons and the medulla. Oh my gosh, there's a piece of cauliflower in this person's head. Is that a piece of cauliflower? No. That's a structure called the cerebellum. Okay, we're going to talk more about the cerebellum in a bit. So again, if you want a really nice picture of brain structure here, the MRI is the tool for you. All right? What's our final tool? Well, our final tool is the one that lets us have our cake and eat it too, because it lets us have information about both brain structure and brain function. So this is really, if possible, probably the best of the techniques that you can use as a neuroscientist. Brain structure and brain function, combining the benefits of both the PET scan and the MRI. Here's the most exciting thing of all. I can't see on list of participants if any of my SI leaders are logged in right now, if you guys are. Or if you've been to an SI session led by Shyla, this is an fMRI of one of your SI leaders' brains. I didn't steal this from anywhere. Shyla emailed it to me and said, Dr. Lemekis, I've been involved in the study up at UCSD where I go in every few months or once a year or something, and they're looking at fMRIs over time. And she goes, I got this really cool letter from them that's got pictures of my fMRI. Can I send it to you? I'm like, absolutely. I'll stick it right into the slide. So here is the cauliflower piece. That is the cerebellum in Shyla's head. We've got some activation in the back of the brain here where she's probably looking at some visual information. It looks like with this frontal lobe activation, she's focused in on some task that they have her doing. So we get both the nice picture of the underlying structures here. Here's her corpus callosum. Again, here's the cerebellum. But we also get the lighting up here, showing us areas that are more active as she's processing different stimuli. Okay? And the fMRI is actually tracing oxygen utilization rather than glucose. The PET scan is tracking the glucose burning up of that fuel. The fMRI is actually looking at oxygen in terms of how it works. All right? Questions on those 5 tools of the neuroscientist? Do they make sense? Is there anything in there that you don't really have a good grasp of yet? All right, no hands are popping up. Let me just show you guys a nice summary of this from your textbook. This is infographic 2.3. I don't have the page number written down, but you can certainly track this down pretty easily. It's a list of ways to study the living brain. So obviously they're not going to be talking about postmortem brain specimen here, but we've got EEGs, PET scans, MRIs, fMRIs. Oh, there's something I didn't talk about. Cool. That's a really good thing to read about in the textbook. Whenever I ask you questions that come just from the textbook, this might be one where I ask you a little bit about a CAT scan and maybe what it tells us or doesn't tell us. So take a look at that. I have been talking way too much, so what I'm going to do for a second is push a couple buttons and make that little check mark up above the list of names become an ABCD multiple choice scenario. Good, I've done that. Take a look at this question. I'll give you guys a minute to think your way through it, and then we'll discuss it. Okay, I'll be back in a minute. Let's take 5 more seconds, guys, and wrap this one up. Cool, let's see how you did. That is 35 out of 46. That's about a little more than 2/3 saying, yeah, this is an MRI. Tell me in the chat box, what made you think that this was an MRI? Not that I'm aware of, Chase. More than anything, it would probably be cost. That's the issue. Still picture, we've got structure, we don't have function. Okay, good. You guys have got the idea here, right? How did you eliminate EEG? How did you know it wasn't an EEG? Because EEG tells us something about function, right? Yeah, pictures were taken. It's not anything about waves, it's a brain imaging device, right? So we can eliminate things and say, okay, so what we've got is they're not sure about functioning, so that it can't be a PET scan because that's functioning, and a functional MRI is functioning. So we can get to the correct answer via process of elimination here, right? That's a great strategy for attacking these sorts of multiple choice items. What can you eliminate, and then what does that ultimately leave you with? Okay? Good. All right. Eris has a question. You can type that right into the chat box. The seem as like a lie detector? Not really, no, because the lie detector scenario would be more like some of those psychophysiological measures that I mentioned at the end of class on Tuesday, right, where you're measuring things like heart rate, blood pressure, skin conductance, the sweating response, those sorts of things would be part of that. Okay, the EEG again is looking at what's going on in your brain at the level of the overlying cortex. Again, if you're studying sleep, if you're studying something like epilepsy, the EEG might be a good tool to use for those sorts of purposes. And Gabriel. Yeah, brain. It totally cannot be EEG because EEG is not an imaging device, right? So she underwent a brain imaging technique where we're taking a picture of the brain. So that's what eliminates EEG. Does that make sense? Because with the EEG, we're not getting any kind of picture of the brain. We're getting this recording, right? So it's not a brain imaging device, right? It is, I'm sorry, it is an image. It's a brain imaging technique, which means it's not an EEG because EEGs are not an imaging technique. All right, we stumbled all over that, both of us. No problem. All right, don't forget to lower your hand so you don't get really a tired virtual hand there. All right, let's turn our attention now to this review of the major parts of the brain. So we're going to spend 20 to 25 minutes taking a look at 7 major brain structures. And as we go through these, I'm going to tell you, like, the hippocampus plays a role in X, or the pons does this. What I want you to understand is that I'm going to give you their major functions. That doesn't mean that they don't also contribute to other aspects of functioning. And then the other thing, from the functional end, when you're looking at something like memory or emotion or visual processing or whatever, most of those interesting aspects of our functioning are governed by multiple structures. There are networks that contribute to them. So it's very rare that there's one structure that does one and only one thing. It's also rare that there's one aspect of behavior that's controlled by one and only one structure. Okay, so things are going to be generally a little more complicated than I depict them. What I'm trying to do is simplify it without making it untrue. Okay, that's really my approach here in the bio section, is to try to strip things down to the basics, but not in doing so make it be the case that I'm telling a story that is incorrect. Okay, major parts of the brain. We're going to do medulla as number 1. We're going to do the pons as number 2. On the next slide, we're going to do the cerebellum as number 3. The thalamus up here is going to be number 4. And I'm going to let you guys not worry about the reticular formation for right now. We've got enough major parts of the brain. So the medulla is sitting right here at the base of the skull. In the girl with this conveniently transparent skull, we can see the medulla right about there. And the medulla is the structure that is the built-in structure controlling basic life support functions. Okay, so the way I think about the medulla is it's the built-in life support system within the brain. What do I mean, life support? It plays an important role in vital body functions like heart rate, breathing, blood pressure, and blood circulation. Let me say all 4 of those again. Heart rate, breathing, blood pressure, and blood circulation. I like to say that there are no unimportant parts of the brain, but obviously the medulla is super important, right? What happens if you damage your medulla? If it's the life support system for your body and you damage it, what happens? You dead, right? Okay, so the good news is it's located in a place where you're not likely to damage this life support system by tripping as you walk across campus and you stumble and bang your head and then you end up dead, right? It's buried in and up through the back of the roof of your mouth and in and up from the back of the base of your skull. Okay, so if you were to able sort of connect the dots between the back of the roof of your mouth and up from the base of your skull, that's where the medulla is. To damage it, you have to do something incredibly dramatic, right? What killed Dale Earnhardt Sr. was a crash where he went from about 180 miles an hour to 0. And your head flies forward, and your head snaps back, and if you break the base of your skull at that high of a speed at impact. That's what could damage the medulla and kill you. Okay, so that's brain structure number one, keeps you alive. Number two, the pons. The pons plays a role as a bridge. Okay, pons is actually the Latin word for bridge, and what it bridges is higher and lower levels of your nervous system. Okay, the bridge between higher and lower levels of your nervous system. Thank you guys for chiming in there. Okay, so we're sending messages via the pons, maybe from the spinal cord up to the overlying cortex. Okay, or maybe the cortex is sending a message via this pons bridge down to the spinal cord. Okay, so that's the primary role of the pons, to function as a bridge. It also has neurons that are involved in sleep and dreams. In fact, when we get to the section on consciousness and we talk about sleep and dreams, a little bit after test number one, we'll come back to the pons, because the pons is implicated in a biological account for dreams. It's called the activation synthesis model, and the activation part of that model really comes from the pons. It's about the pons sending messages up to the overlying cortex while you're dreaming. That seems to be something that's pretty consistently shown in research on why we dream. Okay, so we've got the medulla, the pons. The third structure I've got on the next slide, it's the cerebellum. Here it is looking up from down below. Remember on our nice MRI slide here, we could see the cerebellum right here, this cauliflower-looking structure at the base of the skull. What's the cerebellum going to be all about? Well, the cerebellum, because of the obligatory picture here of David Beckham, is a structure that is involved with muscle movement, coordination, and balance. And that's why we've got a picture of Beckham on the side, right? Or I could have a picture of a dancer, a figure skater, a gymnast, right? All of those would be individuals for whom we would expect that there would be a normal looking, or maybe even better than normal looking, and well-functioning cerebellum. Because all those activities, playing soccer, dancing, figure skating, doing gymnastics, all rely on having really good skills in terms of muscle movement, coordination, and balance. I'll also mention here that the cerebellum is one of the first structures negatively affected by alcohol when you drink. This is one of the first structures that shows some kind of impairment as a function of the alcohol intake. Interesting. What have you seen when people drink that would be connected with the cerebellum here? What do? No coordination. They're stumbling around. They're fumbling. They're clumsy. They're tipsy. This is why, folks, that the police officers, if they stop you and they think you're possibly intoxicated, will have you do tasks like walking heel to toe in a straight line, and other tasks that rely on your abilities to do muscle movement, coordination, and balance sorts of behaviors. If you can't do those things well, and you're showing impairment in those domains, the logic here is we can reason back that alcohol might be something that is leading to that impairment, right? So something kind of interesting to think about. So I guess in terms of the numbering sequence here, this is going to be cerebellum as number 3. So we got medulla as the life support system, the pons as the bridge from higher to lower levels of the brain and vice versa, the cerebellum for muscle movement, coordination, and balance. What's number 4? Back to the previous slide. The thalamus is playing a role here that's extremely important. Again, no unimportant parts of the brain, but the thalamus would be probably in my top 3. Because the thalamus, it turns out, is the relay center for sensory information. So relay center for sensory info is what I'm going to put here. And then in parentheses, if it helps you, I'm going to type in the word O'Hare. And you're going to think he's lost his mind. He's talking about an airport when he's talking about parts of the brain. Here's how the thalamus is like O'Hare Airport. The thalamus is where everything other than smell, in terms of our senses, everything other than smell gets routed through the thalamus. So when visual information comes in for you right now, ironically looking at this slide, right, comes in through your eyes, it will get routed through the thalamus and ultimately dumped back here in the occipital lobe of your cortex in what's called the primary visual cortex. Auditory information obviously comes in through your ears as you're sitting there right now. Trust me, this information is then getting routed right to the middle of your head. Right in the middle of your head is where you've got the thalamus, and then the thalamus will dump that information back out just in and up from where your ears are to these temporal lobes. They sort of look like ear flaps that are wrapped around the brain. The temporal lobes is where that auditory information ultimately ends up. So how is this like O'Hare Airport? Well, if you've ever looked at the route maps for major airlines, United is a perfect example, and you look for flights to the Midwest, you're going to fly to, I don't know, somewhere like, somewhere in Missouri or somewhere in Kentucky or something like that. Many of those flights will get routed through O'Hare Airport. In a similar way, sensory information, everything other than smell, gets routed through the thalamus. Smell has a different pathway. We'll talk about that when we get to the section on sensation after test number 1. Okay. Damage to the thalamus usually results in death, even if you don't damage the overlying cortex. If you damage the center that helps you process all the incoming sights and sounds and so forth from your environment, things don't look good for you. All right? All right, let me take a breath and let you guys sit and cogitate for a minute here. Questions about medulla, pons, cerebellum, or thalamus, the first 4 of the 7 major parts of the brain we're going to look at today. It means death. I wouldn't say indefinitely because I'm not sure what that means in terms of death, but damage usually will result in death. Even if you don't, Gabriel, damage the overlying cortex, damage to the thing that lets you process what's coming in from your world usually means you're done for. Okay. Good. Thank you. All right, we've got 3 more structures I want to mention on this slide. They're all the components of what's known as the limbic system. The limbic system, and I'm going to give you a mnemonic for the limbic system, which is, it spells out the word ha. Okay? It spells out the word ha. How so? Well, it's because number 5 here, we're continuing the numbering, is the hypothalamus. Sounds like it's connected to the thalamus in some way. We'll talk about that in a second. The A of ha is amygdala, and the second H of ha is hippocampus. So what do each of these structures do? The hypothalamus sits under the thalamus. That's why it's called the hypothalamus. That prefix has to do with physical location under something else. So the thalamus is in pink here, and orange sitting below it is the hypothalamus. The hypothalamus is involved in regulating various behaviors. Okay, I'm just going to call it regulating motivated behaviors. What do I mean by motivated behaviors? I'll give you 3 examples, and I'll talk about other things that this thing, the structure, the hypothalamus controls. Regulated motivated behaviors would include hunger, thirst, and sex drive, or what we might call reproductive behaviors. So hunger, thirst, and sex drive are mediated by the functioning of the hypothalamus. The hypothalamus also serves to keep a state of homeostasis in the body because it also controls body temperature, blood pressure, and heart rate. Right? This is one of those situations where, well, you said that was the medulla was the life support system. Well, those things aren't controlled by one and only one thing. The hypothalamus serves to keep things steady in terms, again, of body temperature, blood pressure, and heart rate. It also, as I said, controls things like hunger, thirst, and sex drive. If that is not enough, Oh my goodness, let me give you one more here. It is also the hypothalamus that controls the pituitary gland. Anybody know what the pituitary gland is? Yes, Jackson, that's right, for your question in the chat box. Yeah, it's the master gland of the endocrine system. The endocrine system is your hormone center. So this is kind of wild, right? The master gland itself has a master, and the master of the master gland is the hypothalamus. Again, no unimportant brain structures, but good Lord, the hypothalamus looks like it's pretty damn important, right? All right, number 6 on the parade here of parts of the brain is the amygdala. The amygdala helps to regulate emotion and aggression. So I'm going to type in here emotion and aggression, with a major focus for the amygdala on fear and anger in particular. In terms of specific emotions, fear and anger. And if we want to make this really simple, we could focus in on fear. And I could tell you, for example, let's say that you were someone who was at ground zero in New York City on the day of the 9/11 attacks, okay? And if I were to slide you into a PET scan machine or an fMRI machine, and I were to show you some video with sights and sounds from that day, I'm willing to bet a lot of money that your amygdala would be lighting up as you are re-exposed to those stimuli. Okay? Again, because the amygdala, effectively with respect to fear, it stamps things as emotionally salient or significant. This is something to hold on to. You want to remember this one. Okay? That's effectively what the amygdala does. The final of the limbic system structures here, the second H in HA, is the hippocampus. It plays a role in memory and learning. So I'm going to type in memory and learning. And I would, to make this even simpler, focus on the hippocampus as effectively being the save button for memory. The hippocampus, it turns out, is the structure that lets you store new information into long-term, more permanent memory. Okay? If we want to think about our memory as a computer, the hippocampus is a save button that lets you save that file, that experience, memories of that event, over onto a giant hard drive that is your long-term memory. Okay? By the way, one of the characters from movies that you might remember who had problems with this kind of thing would be Dory from Finding Nemo, or some of the characters in 50 First Dates, or the guy from the movie Memento, where they could not store things onto long-term memory, so they're stuck in the present. They effectively lack this hippocampus as the save button for storing information over into long-term memory. All right? So those are the 3 parts of the limbic system. We've got the hypothalamus. It's kind of in orange, sitting underneath the thalamus there. The amygdala is this sort of little almond-shaped structure there in green. And then the hippocampus is actually this sort of wiry structure that wraps around like that, the hippocampus. All right? Questions about either any of these 3 structures or any of the previous 4 that we talked about, beginning with medulla, pons, cerebellum, and thalamus. Told you it was a big parade of these things today, right? All right, I've been talking a lot. I want to be quiet for a minute. And I want to give you another question that will give you a sense of how I might test you on this stuff, right? It's not just long-term memory, Gabriel. It's effectively, it's the mechanism through which information moves from short-term, or what's called working memory, that's what you're thinking about or working on right now, and saves it over into long-term memory. Like if I asked you what was the name of your 3rd grade teacher, that might be stored in long-term memory. The hippocampus is what helps you take that information when you first meet her or him and save that information over into long-term memory. Does that make sense? It's basically the save button. We'll say more about it in the section on memory. All right. Here's another polling question for you guys. I'll give you a minute to think your way through this. I'll be right back. All right, let's do 5 more seconds to wrap this one up. Cool. Let's take a look. 33 out of 42, that's 11 out of 14. That's about 78, 79% of you say this is the medulla. Yeah, why is it the medulla? What is the medulla? It's the life support system, right? That's the most immediate threat. Okay, could you end up dead with damage to the thalamus? Yeah, but the most immediate direct way to cause that would be to have damage occur to the medulla. All right, good. Let's talk about where we are and where we're going. So we've been talking about some of these major structures. So we've done medulla sitting down in here. We've done pons, it's not really listed here. The cerebellum, that cauliflower-looking structure there, muscle movement, balance, and so forth. Okay, that's pretty cool. We mentioned the thalamus relaying messages, sensory inputs other than smell. Here are the three structures in the limbic system. So we've got the hypothalamus, maintenance functions such as eating, endocrine system, amygdala, emotion, hippocampus, memory. Where I want to go next is to look at the overlying cerebral cortex. So our focus now is going to be on the bumpy stuff that we saw back on the slide where we looked at the postmortem brain specimen. We have two hemispheres, the left hemisphere over on the left side, the right hemisphere over here. And what we're going to do now is take a look at the four major lobes of the cortex and the aspects of functioning that they're in charge of. We're going to do that with this slide here. The colors are just there to help you. Obviously, these parts of your brain are not these colors. So the four lobes, moving from front to back. Begin with the frontal lobe. As we go over the top of our head, we get to the parietal lobe. We circle around to the back of our head now. We're at the occipital lobe. And then these temporal lobes, sort of ear flaps like I described before, right? Up and in from where your ears are, you have the temporal lobe. Obviously, you have a left hemisphere temporal lobe and a right hemisphere temporal lobe as well. So just to orient you guys with this one, obviously, this is the front of the brain. And then back here where the occipital lobe, we've got the back of the brain. So let's talk about what aspects of functioning these different lobes of the cortex are involved with. And I'm going to tell you on the front end, we're going to spend the most time talking about the frontal lobe because it plays a huge role in some really, really important parts of functioning. So it says here on the slide, a higher level cognitive functions like thinking, planning, and personality characteristics. I would summarize that in this way. The frontal lobe is considered to be the seat of intelligence, reasoning, and abstract thinking. What does that mean? Well, just what those words suggest. Intelligence, reasoning, being logical, thinking through things, abstract thinking, imagining what might be, considering possible what-if scenarios and hypotheticals. What's interesting about the fact that the frontal lobe is in charge of these things is the fact that the frontal lobe is the lobe of the cortex that is noticeably bigger in humans than it is in any other animal. If you compare humans to apes or cats or dogs or rabbits or rats or mice, it's pretty obvious when you do that cross-species comparison that the frontal lobe in human beings is massive in comparison to those other animals. That's where we've got all these things that make us unique as animals. Okay, so intelligence, reasoning, and abstract thinking. Number two, the frontal lobe plays a role in helping us to control movement. Okay, and where it does this more than anywhere else is at the very back of the frontal lobe, this little strip here that I'm zigzagging across. We're going to call that the primary motor cortex. Motor means movement. Primary means the main one. So this is the main movement cortex that we have in the brain. So we see it down below here. It's depicted in light blue. Here's the left hemisphere, what I'm going to often refer to as the motor strip, because it's a strip of cortex here across the back of the frontal lobe. Okay. I want to tell you two things about the motor strip that are important for you guys to keep in mind. Number one, it shows evidence of what we refer to as contralateral control. The primary motor cortex, or this motor strip at the back of the frontal lobe, shows evidence of contralateral control, which means as I'm sitting here right now and I raise my right hand, like I'm going to ask a question, right hand is raised, which hemisphere is controlling the movement of my right arm? Is it left hemisphere or right hemisphere? It's the left hemisphere, right? So contralateral control means left hemisphere controls right side movements on my body. The right hemisphere of my brain controls movement on the left side of my body. Okay, that's cool. The other thing I want to mention here is that there's evidence of what I refer to as the proportionality principle. It's a pretty simple idea. It boils down to this. There is proportionally more brain space dedicated to parts of our body where we can do more and more complex movements. I'll say it again. There is more brain space devoted to parts of our body where we can do more and more complex movements. So we can do lots of different movements and facial expressions, so we have proportionally more brain space dedicated to our face or our hands than we do to our trunk or our hip, right? A tiny little bit dedicated in our brain to hip and trunk, a lot of brain space dedicated to face and hands and fingers and thumbs. Make sense? So proportionally more brain space dedicated to parts of our body where we could do either more movements or more complex movements. Okay? That's the motor strip again at the back of this frontal lobe. Any questions on those two aspects of this primary motor cortex? Great. Okay. Those are the first two aspects of functioning I want to mention regarding the frontal lobe. Number 3 is going to be that the frontal lobe possesses what we refer to as executive control. What do we mean by executive control? We mean the ability to plan out and then execute, that is, do, a series of steps that's required to complete some task. Right? So executive control would be part of what helps you be able to follow the steps in a recipe or follow the instructions to build whatever you just brought home from IKEA, right? Or to plan out what you need to do at the beginning of the semester to get your courses underway. I need to register, and then I need to look at the syllabus and find out the materials that I need, and then I need to go to the bookstore and buy those materials. All of that stuff. Anytime you have to plan out and then complete a series of steps, then you're talking about executive control, and the frontal lobe is in charge of helping you to do that. Okay. Finally, the frontal lobe is what is involved with helping us to inhibit impulses, to keep us from doing or saying things that might get us in trouble, what we might call inappropriate responses. So you never want to say the word bomb anywhere near an airport. You never want to yell fire in the movie theater, unless these things are really happening, right? You never want to yell out in class, "This is the most boring lecture ever, would you just be quiet?" Even if you want to, right? So effectively, in inhibiting impulses, the frontal lobe functions as the brakes on the car. It helps us to slow down, to think things through, to consider consequences, and then to act appropriately. Okay? Now, having said all that good stuff, right? Slow down, think things through, consider consequences. I'll mention that the frontal lobe is not fully developed during adolescence, and in fact, it continues to develop through our 20s until about age 25 or so. So, does this explain things that go on during your teenage years? Are there times in your teenage years where, looking back, you made some decisions that probably weren't in your best interest? And maybe you made those decisions because you didn't think things through all the way. Okay? So yeah, this filter idea, Kelly, is another way to think about it. But more than anything, I like the idea of brakes on the car, right? Right? If I'm a teenager who has an impulse, you know what, dude, I'm gonna get on this. Yeah, here's what I'm gonna do. I'm gonna take my skateboard up on the roof, and I'm gonna skateboard down the side of the roof and fly into the pool. And that was like a couple of broken legs and a broken arm ago, right? So again, continues to develop through our 20s, and again, might explain why we've got some of the craziness that we get with teenagers. By the way, one of the ways that we came to learn the role that the frontal lobe plays in inhibiting these impulses like this is from the case study of Phineas Gage. I mentioned him last week when we were talking about case studies as an approach to descriptive research. Remember, Gage was the guy who had the tamping iron working on the railroad all the live long day. There's a blast that blows through his skull, through his left cheek, and up and out through the top of his left frontal lobe. I mentioned last week that his doctor said Gage was no longer Gage. Well, what he really meant was that his personality changed in a really significant way, and he wasn't very good anymore at inhibiting impulses. Okay? So if he didn't like your haircut, he'd be like, what the hell were you thinking? Right? Or he would curse and swear to a much greater degree than he ever had before. Okay? So that's how we know some of the things that we know about the frontal lobe. The parietal lobe, number 2 here, is in green. It says integration of sensory information like touch and temperature. I'll make this simple. This is about processing the sense of touch more than anything else. There's some other things to be added, but this is intro. And our sense of touch is really specialized on this strip here at the front of the parietal lobe and adjacent to the motor strip at the back of that frontal lobe. We call this our primary somatosensory strip. So just like we have the motor strip here at the back of the frontal lobe, we have this lime green somatosensory cortex, the somatosensory strip, at the front of the parietal lobe. It also has contralateral control, which means what? Well, if you have a blindfold on and I stroke the palm of your right hand with a feather and ask you to tell me what it is that I'm touching your hand with, well, you're processing that sense of touch in your right hand in which hemisphere? Right hand touch processed in which hemisphere? Left. Good, and vice versa, right? If I squeezed your left arm and you felt that touch, that would be right hemisphere lighting up in the PET scan or the fMRI, something like that. So we've got contralateral control, and then we've got proportionality again. More brain space dedicated to parts of our body that are more sensitive to touch. Without getting overly graphic here, right? There's more brain space, for example, dedicated to your genitals than to, like, your forearm, right? Nobody gets really excited when you touch them on their forearm. Other places, it's a different story, and I need to stop talking now. So we get the proportionality principle here in play also for this somatosensory strip, again, where we have the sense of touch from head to toe. Proportionally more brain space for parts of our body that are more sensitive to touch. Got it? Okay, good. That is what's going on here in the parietal lobe. Occipital lobe, we've mentioned a couple times here. Let's just explicitly point it out. It's where we process visual information. So I think about occipital lobe and I think of vision. The occipital lobe receives information from your eyes. Obviously, that information gets routed through the thalamus and then analyzes that information to give us sight. If you've ever received a pretty significant blow to the back of your head, did you notice anything when that happened? In terms of vision, a blow to the back of the head might make you do what? Did you ever see stars? Yeah, Gabriel's got it, right? So you might see stars. You're sort of manually stimulating this visual cortex. You don't want to do that. That's not good. Okay. I would also note here that you could end up with partial or even complete blindness if you were to have a stroke and damage the visual cortex here in the back of your head. So you could have perfectly functioning eyes in the front of your head, but you could end up with what's called cortical blindness because of damage at the level of this overlying cortex. So vision in the occipital lobe. And then finally the temporal lobe. Again, we've got a left temporal lobe, a right temporal lobe here for hearing and language comprehension. Basically, the temporal lobe integrates sound information. So the temporal lobe does for hearing what the occipital lobe does for vision. Okay, so to keep this really, really simple, I'd say occipital lobe is vision, temporal lobe is hearing. And then you're done with those. You're going to spend more time than anything else getting your head around the aspects of functioning the frontal lobe is involved with here. Okay? If you want to see the visual and auditory cortices together here, we've got this slide with the girl with the nice transparent skull, visual cortex back here in the occipital lobes, auditory cortex here in the temporal lobes. We're seeing it in her left temporal lobe. There's obviously an auditory cortex. If we spun her around and looked at her right hemisphere, we'd see the auditory cortex in her right temporal lobe as well. Okay? Questions about these major lobes of the cortex before I just blast ahead here? Does that make sense? Okay, I got two more things I want to do with you. I want to talk briefly about language in the brain. It's actually going to circle back to a point that we made when we looked at the postmortem brain specimen at the beginning this morning. And then I want to do a final polling question to give you, again, a flavor for the kinds of test questions that will come from all this material on the major parts of the brain. So let's take a look at language in the brain and be a little more explicit about something that we were alluding to earlier. We've got an area called Wernicke's area here. It's kind of at the top rear part of the temporal lobe. Wernicke's area is the structure here that plays a significant role with respect to language comprehension. Okay? Comprehension. So being able to understand what people are saying to you. Again, as you're sitting there right now and listening to me, it would be Wernicke's area that will be lighting up in the PET scan or fMRI machine. Broca's area, in contrast, which sits at the back bottom part of the frontal lobe, is a structure that's involved with language production. So I'm going to type in here, language production. So speech production, for example, what I'm doing right now. I have to move my mouth and my lips and my tongue and my vocal cords. And it kind of makes sense that Broca's area is there because the motor strip that helps us to move things sits right here on the front side of that major fissure between the frontal lobe and the parietal lobe right behind it. So that motor strip at the back of the frontal lobe sits right next to the speech production area. Okay, where these things get interesting, and how I usually like to ask questions about this kind of stuff, is I'll ask what happens if you were to damage one of these areas. So, what if you were to damage Broca's area? What could you not do? Yeah, you wouldn't be able to speak. You wouldn't be able to talk. What if you damage Wernicke's area? Yeah, these are just in the left hemisphere. Good question, Ariana. These are just left hemisphere. Broca's and Wernicke's. Yeah, if you damaged Wernicke's, you couldn't understand what people were saying. Now, this is a really wild scenario. Imagine you damaged Wernicke's, but this Broca's area here was just fine. It was intact. It wasn't damaged at all. You wouldn't understand what people were asking you, but you could still produce a response. Tell me what kind of a response you would typically give if you had that kind of a scenario, where you didn't understand the question, but you were responding anyway, because you've got Broca's area intact. Yeah, it would be unrelated. It wouldn't even make sense to the question. Caitlin does a nice job there, right? It wouldn't make sense. So, like, I would ask you, like, well, what are you planning to do this weekend? And you'd be like, I really love the color blue. It is fantastic. It makes me feel so, what? Why are you talking about blue? Right? I'm asking you about what you're planning to do over the weekend. So it's that nonsensical kind of rambling speech. Again, when you've got damage to Wernicke's, but not to Broca's. We're going to come back to this later on. We talk about development. We'll have a little segment on language development. We'll talk about what's called expressive language and receptive language as well. Sign language will be mediated by different parts of the brain. Adrian's asking if Wernicke's area is damaged, so that's language comprehension. Can you still read and understand? It would depend. Adrian, it really would depend on how localized and specific the damage was. So all of these things, again, also involve networks, so it's not just one and only one structure. So it would depend on the specifics of the case. But many times what happens is in the aftermath of a stroke, what we see is what's been knocked out, what you can't do, and then we sort of deduce what the underlying damage is likely to be. Okay, we're probably going to confirm that with an MRI or an fMRI, something like that. But it's hard to say, exactly how these things would work out until you knew the exact specifics of a given case. Make sense? I've got a final question for you guys. Let's do one last polling question. If you want to make a laboratory cat twitch its whiskers, that is, move its whiskers, which lobe of the cortex would you want to stimulate to make that happen? All right, how about 10 more seconds before we wrap this one up? Let's take a look. How'd you guys do? All right, 22 out of, what the heck is that, 37. I don't even know what that is on a percentage basis. It's less than 2/3 are saying it's frontal. It is frontal. Why is it frontal? Think about what goes on with the frontal lobe. We've got the motor strip, right? Twitching your whiskers is about moving your whiskers. The movement control is mediated by that primary motor cortex here at the back of the frontal lobe. The strip right here, or if you like it better on this slide, where I zigzagged across it with this summary of the different lobes of the cortex. Does that make sense? Anybody still confused about why it would be frontal lobe here in this case? All right, cool. Here's where we're headed next. On Tuesday, when we get together face-to-face, we are going to talk about the corpus callosum. We've got two views of it here on this slide. This view is what's called the sagittal view, where we split the brain down the middle. This is a horizontal view down into the brain. And this corpus callosum, as we're going to learn on Tuesday, is the bridge between the two hemispheres. It's the bridge that lets information that is initiated in the left hemisphere be shared with the right hemisphere, and vice versa. So, for example, if we wanted to coordinate movements and do jumping jacks and not look like a maniac because nothing is coordinated, we're going to need to dump that information back and forth between the two hemispheres. The mechanism through which we do that is this bridge, this band of fibers known as the corpus callosum. So we'll talk more about that on Tuesday. And most interestingly, what we're going to look at is what happens if you cut that and you don't let information move back and forth between the two hemispheres. That's where it's going to get really, really interesting. Gabriel, final question. And if anybody else has a question, just raise your hand. Difference between frontal and parietal. Yeah, it's just basically right at that fissure is where you see the separation between those two. So the frontal lobe does all these things. The split is right here. At the back of the frontal lobe is that motor strip, and then at the front of the parietal lobe is where we've got that somatosensory strip about the sense of touch. Okay, so there is this central fissure, this split here, that separates those two. I don't want to get too detailed and make you guys identify and be able to label something like that. I just want you to understand generally where things are, right? Frontal lobe is well in the front as we move towards the back of the head going over the top. Parietal lobe is next, occipital in the back, and then temporal around the sides. If you've got that much, that's good enough. All right? Awesome, you're welcome. Any final questions on anything regarding the major tools of the neuroscientists or these major parts of the brain today? If you showed up late and you didn't get a list of the breakdown number of questions for test number 1, there it is. We're going to finish up, and remember, when we're done, I need everybody to exit out so we can make the recording available to your classmates. I will have online office hours at 11. I'll make that link available through the button on Blackboard that says Tuesday online office hours. And we'll go from there. I'm sorry, Thursday online office hours. All right, I'm done. Obviously, my brain is fried. We will wrap up here. I'll see you guys not first thing in the morning, 9:30 with you guys on Tuesday. Don't forget, Learning Curve quizzes for Chapter 1 are due Sunday night at 11 o'clock. Get in there and get them done. You're welcome, Marianne. Have a good rest of your Thursday. I'll see you guys Tuesday. Bye, everybody.